All right, so I want to make sure to get through this stuff today, so I'm going to go ahead and move on. What I talk about over the next 15 or 20 minutes, you should be able to fill in the rest of the activity. Okay, so we're now at a point where we want to think about how these two pathways are regulated, right? And in terms of sensing conditions in a cell, this makes sense. It genuinely makes sense if you just take a second to think about it, right? We know that a buildup of ATP means that there's lots of energy in the cell. And when we talked about regulating glycolysis, we said that that inhibits enzymes in glycolysis, right? It inhibits PFK. It inhibits pyruvate kinase. That buildup of ATP is inhibitory because it means the cell already has plenty of ATP. The reverse has to be true then for gluconeogenesis. If we have a buildup of ATP, we want to use that energy for something useful. And one of the roles of gluconeogenesis is to divert metabolites back through glycolysis so we can store energy in the form of glycogen, store energy in the form of sugar polymers that we can use later. Okay? So if we look here at these two pathways, on the left side we have glycolysis, on the right side we have gluconeogenesis. And we're pointing out here two key regulatory enzymes, phosphofructokinase and pyruvate kinase for glycolysis, and then FBPase, and then our two pyruvate to PEP enzymes for gluconeogenesis. Okay, two key steps. The most important step we're going to see is the PFK and FBPase step, but the PEP and pyruvate interconversion also plays a role. Okay, in terms of metabolites of the cell, this makes sense. For glycolysis, a buildup of ATP inhibits PFK. It inhibits pyruvate kinase. That makes sense because a buildup of ATP means we have plenty of cellular energy. We want to store it, so we activate gluconeogenesis. A buildup of AMP means that we have depleted energy reserves. So this is activating phosphofructokinase. That makes sense. A depletion of energy means activating flux, increasing flux through glycolysis so we can make more energy. That makes sense. The reverse is true for gluconeogenesis. Gluconeogenesis is strongly inhibited by AMP. AMP means cellular energy is highly depleted, very, very low. We're running on reserves. So let's activate pathways to make energy and make sure we're inhibiting pathways that consume energy. Gluconeogenesis consumes energy. Let's stop it. We stop flux or slow down flux through gluconeogenesis by inhibiting FBPase, by inhibiting hydrolysis of fructose 1,6-bisphosphate. Additionally, a buildup of ADP inhibits pyruvate carboxylase and phosphoenolpyruvate carboxykinase and PEPCK, right? So these two enzymes converting pyruvate back to PEP are inhibited by conditions that aren't quite as intense, that doesn't signal energy depletion quite as much, okay? Right, because remember, ADP buildup doesn't necessarily mean that we're at truly deficient energy levels, okay? So we kind of have this graded regulation. So a buildup of ADP inhibits the conversion of pyruvate to PEP. So this effectively inhibits gluconeogenesis. Even more so, a buildup of AMP, which signals even lower cellular energy, will inhibit the FBPase. Okay, so it's kind of like a graded response. Depending upon how deficient the cell is, we will inhibit certain key enzymes. Other things we see here, like citrate inhibiting glycolysis, citrate activating gluconeogenesis, this is another cellular signal, right? So citrate is a metabolite of the TCA cycle. If we have a buildup of citrate, that means there's a buildup of TCA cycle intermediates. So if that's the case, we don't really want to activate glycolysis because we already have a buildup of intermediates downstream. So a buildup of citrate inhibits glycolysis, but it activates gluconeogenesis. The same thing goes for alanine and acetyl-CoA. We can make the same arguments, which we'll explore those more later. Okay, so this feedback regulation, feedback activation or feedback inhibition based on the cellular demand of the cell, the energy demands of the cell makes a lot of sense. Okay, the other molecule that we see on here, this F26BP, this is the one that things become, I think, really interesting, but also pretty complicated pretty fast. Okay, so I'm going to try to work through this slowly, and I'll say it several times if I have time, so that hopefully it kind of clicks. Okay, so this molecule, fructose 2,6-bisphosphate, it's an isomer of fructose 1,6-bisphosphate, right? It's phosphate on a different position. The idea here is it's activating, strongly activating towards PFK and strongly inhibiting of FBPase. It strongly activates glycolysis and strongly inhibits gluconeogenesis. And the magnitude of this is remarkable. Really, really, really small concentrations of fructose 2,6-bisphosphate aggressively activate PFK activity, right? We're talking about 100 nanomolar. The blue line here is 100 nanomolar, and we see massive activation. Even more so if we get up to 1 micromolar, right? So if we have 1 micromolar of this F26BP around, we have low micromolar affinity of PFK for our substrate. Okay, so it's really active. Really small amounts of substrate, fructose 6-phosphate, will trigger activity of PFK. Okay, strongly activating. The interesting thing here is that the activation by this molecule, fructose 2,6-bisphosphate, can be tempered, it can be dampened by a buildup of ATP in the cell. Okay, so here we're looking at our green line again is 1 micromolar, our blue line is 100 nanomolar of our activator. We see here that as the ATP concentration in the cell increases, the activation is still there, but it's not nearly as dominant. Okay, so ATP is the signal inside the cell that says we don't need this enzyme to be active. Fructose 2,6-bisphosphate is actually a signal that originates from outside the cell. It's a way of communicating the overall organismal demands of energy, communicating inside the cell. Okay, so these two signals, fructose 2,6-bisphosphate and ATP, work together to give the proper coordinated regulation of these metabolic pathways. Okay, the fructose 2,6-bisphosphate signal is much, much stronger, right? We think about these concentrations. We are talking about millimolar ATP to start overcoming nanomolar or micromolar F26BP, right? So the organismal demands really matter. How does this happen? Okay, so this can get, I love that face. So what the hell am I looking at? This can get confusing, and I'm going to try to go through it slowly enough that it makes sense. Okay, so we are starting thinking, all right, so when we eat, right, when we eat something that's sugary, it goes into our blood, right? So the organismal demands are going to boil down to signals that are passed through the blood, okay? So we are thinking about blood sugar here, right? And hopefully when we say blood sugar, hopefully everybody's mind goes to insulin, right? It is all that is one of the main signals of blood sugar. Okay, insulin is the hormone secreted by the pancreas when there's a high blood sugar, right? High blood sugar signals the pancreas to make insulin. Low blood sugar, in contrast, signals the pancreas to make glucagon. These are opposing hormones. Okay, insulin means high blood sugar, glucagon means low blood sugar. And before we even think about anything else here, let's think about what this should mean for glycolysis. If we have high blood sugar, that means that we want to bring that sugar into the cell to be used, right? And it's coming into the cell, so it would make you can make an argument based on what's happening inside the cell, either for glycolysis to be active if we have low cellular demands, or glycolysis to not be active and storing that sugar depending upon the ATP cellular levels. Okay, glucagon if we have low blood glucose, the role of the liver, and this is really important, okay, because this idea is pervasive the rest of the semester. The role of the liver, the role of hepatocytes, is in many ways to make sure the brain and the heart can function properly. It is central in making sure other organisms function right. Okay? So when there's low blood sugar, we want all energy reserves in the liver to be mobilized to send energy elsewhere, especially to the brain. Okay? So the liver is really good at storing glycogen. It can store sugar in the form of glycogen. So when there's low blood sugar, we secrete glucagon, and that glucagon needs to tell these cells to mobilize glycogen, mobilize all possible glucose to secrete that glucose into the blood so we can send that glucose to the brain. Okay? So with that being said, we want glucagon, we want low blood sugar, glucagon signaling to trigger any possible pathway of making glucose. So under these conditions, we want gluconeogenesis to be active, and we want glycolysis to be highly inhibited. Okay? If you can capture that idea, then everything else kind of starts to fall into place. But you have to have this down first. Okay? The cool thing about these opposing signals is that they actually end up with opposing signals in the cell in terms of phosphorylation. Okay? So you may remember from Biochem 1 that insulin triggers the insulin receptor, which is a tyrosine kinase. I saw her mouth it, right? It's a tyrosine kinase. And ultimately, the tyrosine kinases do a ton of things. One of the things that they do is they activate protein phosphatase 1. Insulin activates phosphatases in the cell. Commit that to memory. Insulin activates phosphatases in the cell. Glucagon, so the glucagon receptor is a G protein coupled receptor. G protein coupled receptors function by producing cyclic AMP, which then activates protein kinase A. Okay? Insulin activates phosphatases in the cell. Glucagon activates kinases. These hormones are secreted under opposing conditions, and they activate opposing enzymes in the cell. That makes sense. Insulin activates phosphatases. Glucagon activates kinases. Okay? So how does all this work? So this molecule fructose 2,6-bisphosphate, right? This is the key activator of glycolysis of PFK. It's also a potent inhibitor of FBPase. Okay? It is synthesized from fructose 6-phosphate by this enzyme PFK2. Phosphofructokinase, we know its role is to convert fructose 6-phosphate to fructose 1,6-bisphosphate. An isozyme, an isoenzyme of PFK is PFK2, which creates an isomer of FBP. It creates fructose 2,6-bisphosphate instead of fructose 1,6-bisphosphate. Okay? This matters because if this enzyme is active, we make fructose 2,6-bisphosphate, which activates PFK, which activates glycolysis, and strongly inhibits gluconeogenesis. It strongly inhibits FBPase2. These two enzymes, so PFK2 and FBPase2, the enzymes that are responsible for making F26BP or degrading F26BP, are part of the same enzyme. Okay? One enzyme, two activities. And which activity is active, which enzyme active site is active, depends on the phosphorylation state of the enzyme. Okay? When a phosphate is added, it activates one domain. When the phosphate is removed, it activates the other domain. And this is the link to insulin and glucagon signaling. Okay? I'm going to run out of time. It's okay, we'll come back to this on next week. All right, so what we're looking at here is a map that I made that kind of shows the linkage with everything. Okay? In the middle, wrong button. In the middle, we have the two different active states of our key enzyme, right? So we have this enzyme that has a PFK2 domain, a kinase domain, which makes fructose 2,6-bisphosphate, and it has an FBPase2 domain, which degrades F26BP. And what I want you to notice here, the key thing here is that which domain is active depends on the phosphorylation state of this enzyme. When this enzyme is phosphorylated, it activates the FBPase domain, which degrades fructose 2,6-bisphosphate. When the phosphate is there, F26BP gets degraded. When the phosphate gets removed, fructose 2,6-bisphosphate gets made. When F26BP is present, we inactivate FBPase, we inactivate gluconeogenesis, and we activate PFK, which activates glycolysis. Okay? Try to show it in a couple more ways here. So here, I've highlighted only what's going on when blood glucose levels are high. Insulin's being secreted. Remember, insulin secretion means phosphatase activity. Phosphatase activity removes the phosphate from our central enzyme, activating the PFK2 domain. The phosphatase secreted that's produced when insulin is secreted, its role is to remove the phosphate from this enzyme, which activates the kinase domain, right? That's counterintuitive. The phosphatase activates the kinase. But when the kinase is active, we are making F26BP. Gluconeogenesis is inhibited, glycolysis is activated. Okay? The opposite's true when we have low blood sugar. When we have low blood sugar, this means glucagon is activated, glucagon secreted, and glucagon leads to kinase activity in the cell. We activate protein kinase A. Protein kinase A is responsible for removing the phosphate from our core enzyme. That makes the phosphatase domain active, which actively degrades fructose 2,6-bisphosphate and makes F6P. We no longer have this activator of PFK, so gluconeogenesis is activated and glycolysis is inhibited. Okay, I told you all are going to hate this, but once you wrap your head around it, it does make sense, and it's really, really freaking cool. Okay, so we're going to stop there. On Tuesday, I'm going to review this one more time, and then we're going to move on, and we're going to dive into thinking about other sugar metabolism, and then as time permits, we'll get into the TCA cycle. I'm at least one full day behind, maybe a day and a half at this point, but that's okay. Okay. Next week, we will revisit the schedule and kind of like see where the next exam is going to be. But I would rather take our time and go through the stuff so that everybody's grasping it. We're going to review this. Please take some time to look over it this weekend so that I'm not talking to people that gives me these faces anymore. All right, Kenny?